So hi everyone, welcome to our Serie R for Bioinformatics and to our workshop Introduction to Genomic Ranges and Plyranges. Today we have the pleasure to have with us Dr. Stephanie Hicks. Dr. Stephanie Hicks is an assistant professor in the Department of Biostatistics at Johns Hopkins Bloomberg School of Public Health. She's also a faculty member of the Johns Hopkins Data Science Lab, the co-host of the corresponding author podcast, and the co-founder of IRanges Baltimore. Thank you, Stephanie, for accepting our invitation. It's a great pleasure for us to have you today. And I think we can start. Should we make a presentation of IRanges Tunis, or shall we start with the workshop because the time is really limited? So I think we start with the workshop. Let's start.
Let's start with the workshop.
Let me quickly share my screen. Can someone confirm that you see slides?
Yes, yeah. Yes, we are seeing your slides.
Thank you. I was just checked before diving in. Well, first of all, thank you so much for this invitation. I was really excited to receive it and to get to know your community. As Hedda said, that I'm a co-founder of the IRanges in Baltimore, so this is a group that's near and dear to my heart. And then also being able to talk about the analysis of genomic data is also very near and dear to my heart, so it's hard to resist such a topic. Today I'm going to talk about two packages in the Bioconductor ecosystem. So if you're familiar with CRAN, Bioconductor is kind of like a cousin or a sister to CRAN for our packages that are developed. Oh, there's some echo. Oh yeah, okay. Our packages that are developed for biological data. And so GenomicRanges and plyranges are specifically the two. And the reason I'm interested in presenting these are GenomicRanges and GenomicRanges objects are really the base infrastructure of almost all Bioconductor data. So the way that genomic data are stored on a computer are more or less in what's called a GRanges object, and you can manipulate those objects and do cool things with them with the GenomicRanges package. So if you're interested in learning about how to store data in our Bioconductor, how to work with these data, this is the package that you want to start with. Plyranges is really cool because plyranges allows you to, similar to the way we talk about the tidyverse, being able to make code more human readable and being able to easily pipe output from one function into the input of another function. It enables us to do that with genomic data analyses. So today I'm so excited to present both of these packages to you, and then at the end there's a little demo. I'll state at the beginning, my slides are available on stephaniehicks.com/talks if you just scroll down to today's meetup. And then inside of the slides, there's a link to an actual R script with a demo if you want to follow along with it at the end of the talk. And please feel free to just see the chat box. I don't know where it went. So if there is a question, please feel free to open up your mic and ask. All right. So I come at data analysis from a genomic perspective, and I thought I would briefly talk about what are genomic data, and specifically what does it mean when we talk about we're going to generate some genomic data and then we're going to analyze it. I thought it would be useful for those that are not familiar with genomic data to briefly cover that. So at the bottom of the screen is a piece of DNA. So these are the A's, the T's, the C's, and the G's. These are 4 nucleotides that make up the DNA, and every cell with a piece of DNA that we want to sequence. And it could be a piece of my DNA if we want to sequence my genome, for example. And then what we do is we take it and we fragment it up into tiny little pieces. And then from there, the sequencing experiment produces what are called sequencing reads. And these are very short length reads of maybe 50 base pairs, sometimes 200 base pairs. More recently, you can get long read sequencing, so like millions of base pairs, but typically they're very short reads. So we get these sequencing reads as the output from a biological experiment. And each biological experiment can contain billions of reads, lots and lots of reads. And the question is, what do you do with these reads? Because we originally had this long piece of DNA that we're interested in sequencing. Now we've fragmented it up into tiny little pieces. How do we figure out where each read came from? So I'm going to tell you how we do that. So we take a reference genome, such as the human reference genome or the mouse reference genome, depending on what species it is. And there are things called gene bodies. So there are these pieces of the DNA that code for things such as proteins. And we're interested in them because they're functionally important. So if a piece of DNA gets transcribed into a piece of RNA, and then an RNA gets translated into a piece of protein, that protein is going to go on and do something. It has a task. And so it's functionally relevant to the cell. And so we're interested in looking at reads that come from genes. And so I'm depicting here in a schematic gene XYZ that is coming from some human reference genome. And I talked about these reads before that come from a sequencing experiment, say my genome, for example. So these are reads that came out of a sequencing experiment, and we have to figure out where do they come from relative to some human reference genome, for example. And so we basically do pairwise similarities. We say, what's the likelihood that this read comes from this particular region of the genome? And we're just comparing the A's, the T's, the C's, and the G's, and we're saying, like, how similar are they? So there are many algorithms that do that. So let's just assume that we've run those algorithms, and these were where the reads come from. So each little bar there represents a particular read from the schematic before. So what we do is we just sum them up. So we say all of these reads map or come from gene XYZ. And in this case, there are 23 reads that come from gene XYZ. So what do we do with that number 23? We just record it in a matrix. So for gene 1, for sample 1, for Stephanie's sample, for example, we record that there were 23 reads that came from gene 1. You can repeat this. Say for everybody here on the call, we can count the number of, we can perform a sequencing experiment, sequence our genome. We can count the number of reads that map to gene 1, for example. And then we can redo this for every gene in the human body, for example. And in the human body, there are around 20,000 protein coding genes. So G here could be around 20,000. So it's a pretty big matrix. And this is the matrix that we're interested in analyzing. There's also this other matrix that I like to call metadata. You could think about it as really, really important bookkeeping. And I'm going to talk a lot about today about this, this matrix here on the left. But for each gene, say that comes from the human reference genome, we have to know which chromosome did the gene come from and what was the start position. Did it start at base 1, base 2, base 3, base 4, or so forth? And then where does it end? Where does the gene body end? And so in this case, gene 1 started in chromosome, it comes from chromosome 1, and it started at position 10, it ends at position 500. Gene 2 starts at, it comes from chromosome 1, starting at position 400, and then ends at position 882. So it's very important bookkeeping because you want to be able to figure out where the gene comes from in a reference genome. But as I mentioned before, this is the data that we typically analyze. So for example, just to give you like a brief example, we could ask the question, what's the average expression in the first 3 samples from gene 1? So if you just take the average of 23, 10, and 15, and you compare that to the average of the expression in the last 3 samples, 3, 0, and 1, and you ask, is there a difference in mean expression between the first 3 samples, maybe that's a healthy group, and then the last 3 samples, maybe that's a disease group of sorts or a case group of sorts, and you want to figure out which genes are different, say, between two groups. That's genomics kind of in a nutshell. So how can we analyze these data? The way I do it is with Bioconductor using the R language. There are many different languages out there that you can use to analyze genomic data. I thought I would just briefly touch about Bioconductor because it's an important component to this talk. So I know you've heard from Martin Morgan and Mike Love already, but I'll just restate this. Bioconductor is an open source and open development software project for computational biology. Open source meaning anybody can read and modify the underlying code, while open development means anybody can contribute to the code. It began in 2001 with, by Robert Gentleman and others, and now it includes like 12 full-time core developers to build and maintain it. It has over like 2,000 packages in the project. It has a lot of big priorities that focus a lot around reproducible research with high quality documentation. Okay, so because I talked about Bioconductor and because this kind of like ties into the tidyverse, I thought I would showcase a really cool package that I've discovered recently called BioC Package Tools. So what it does is in one line of code, so if you load this package, BioC Package Tools, if you run the BioC download stats, it can give you a tibble or a tidy data frame of download statistics for all Bioconductor packages in the project. So every row contains one observation associated with the name of a package, the month and the year, the number of distinct downloads, the number of distinct IPs, the type of repo it is, which I'll talk more about that in a second, and then the date. So because it's a tidy data frame, you can basically immediately apply functions from the tidyverse to it. So here, for example, you can take the package data frame and you can filter for all the packages in the year 2020. You can select for the column repo. You can group by it, and you can essentially count how many of each type of repository is there. So you'll see that Bioconductor has 3 different types. There's something called annotation data, experiment data, and software. So annotation packages basically streamlines that bookkeeping that I talked about before, which is I think what Martin Morgan talked about at your earlier meetup. And then experiment data packages, they contain process data, really useful for teaching. And then software packages are the ones that I'm going to talk about today. So you can quickly create ggplots with software, with the tidy data frame from this package. So you can quickly figure out like the fraction of, or the number of packages that have been contributed over time and so forth. All right. So one of those packages is called GenomicRanges. And as I mentioned at the start of the talk, it's really a foundational package for Bioconductor. It is the, I would say, the standard data structure that Bioconductor uses to store genomic data. So I thought I would give you an example of what is a genomic ranges at first. So here we have a piece of chromosome, so that chromosome 1, for example, that I was talking about. And the way we're interested in recording gene bodies, for example, or gene regions, or genomic regions in general, doesn't have to be a gene, is we need to record information such as, for a genomic region, where does the genomic region come from? Which chromosome? What is the start position? What is the end position? The strand, I'm just going to ignore for now. It's like it's something about biology. You can read genes, or you can read genomic regions in 2 different directions, the positive or the negative, but we'll just ignore that. And then you can store like metadata, such as the name of the gene or an ID for a gene or a genomic region, and then a score. So the score you can think about as like any metric you want to record, such as the number of reads that map to that genomic region, like that number 23 that we talked about before. Or it could be a totally different piece of information, but it's just some piece of information you want to record from that genomic region. And this is, this object or this class, the GenomicRanges class, follows the tidy principle. So each row here corresponds to one interval, while each column will represent a variable about that interval, And generally, each object will represent a single unit of observation, such as gene annotations and so forth. So this follows the tidy principle, which is really cool. And I'm going to show you why. Next, I thought I would briefly show you just how we create a GenomicRanges object in Bioconductor. So you load the GenomicRanges package, and then you can use the function GRRanges, for example, and it has some requirements, some things that are required to create a genomic packages and things that are not. If you look, there are 4 lines corresponding that split the object in half. It's in between the columns strand and gene ID. Everything to the left of that column is required, and everything to the right of that is not required. It's just metadata, whatever you want to store about that genomic range. So things that are required, you need which chromosome did it come from. Strand, I think strand might be actually optional, but because I've gotten away with not recording strand before. And then ranges. So there are things called IRanges. It's a particular, IRanges is very similar to GenomicRanges, but it doesn't require any information about chromosome region or strand. It just needs to know the start, the end, and the width of the genomic region. And I'll give you an example of that in the demo. And then the 2 other columns that I created are just gene ID and score. So it's really easy to create one of these objects in Bioconductor. Things you can do with this object, just right off the top. You can calculate the width of each region. So as I noted here, there is a start and an end position to each genomic region. You can just calculate the difference between the 2 and calculate the width of that region. You can apply base R principles to do filtering, such as using the open bracket notation, where everything on the left side of the comma will filter by rows, and everything on the right side of the comma will filter by columns. So here I'm using base R notation to filter for rows that only have a score greater than 15, for example. So you can apply base R notation. But then the second package I wanted to highlight today is something called plyranges. So plyranges, the goal of plyranges, in my opinion, is to make genomic data analysis more human readable in the same way that I think that dplyr, for example, was created to make generic or broadly applicable data analyses more human readable. And the idea is to define an API. What that means here is basically extend the dplyr API that maps relational genomic algebra to verbs, such as select, filter, join. These are action verbs that you can take upon a particular data set. And so the idea here would be to basically borrow that syntax that was developed for dplyr and the design principles behind it, and apply it to genomic data, or specifically genomic ranges, as they follow, as they are considered themselves a tidy data frame. And so you can also then combine it with things such as the pipe operator from the Moderator package. You can gplots the same way you would with regular tidy data frames and so forth. So I'll give you a quick example, give you a summary of some of the figures, and then there's a demo. All right, so here I'm loading in the plyranges package, and we had that GenomicRanges object with two genomic ranges in it. And instead of using base R notation here, I'm piping the GenomicRanges object into the filter function. And if you're familiar with dplyr, you might be familiar with the filter function, which allows you to filter rows based on a particular criteria. And here I'm filtering for that same criteria. I'm asking for all the rows with a score greater than 15. Similarly, you can then pipe that into another function, such as width. And I can, in 3 lines of code, in a very human readable way, I can take my GRanges object and calculate the width of all the genomic ranges that contain a score bigger than 15. And you can imagine, like, there's a lot you can do when the world opens up to you like this. So it's really exciting. So this is a brief overview of the functions inside of plyranges. The bold ones, summarize, mutate, select, arrange, they actually come directly from, they're borrowed directly from the dplyr API. And then there are a whole bunch of new ones that were developed specifically for genomics. So there are a lot of things that you don't have to deal with in just regular tidy data frames that you do have to deal with in genomics. And I'm going to talk about some of these, but a lot of them are you might be very familiar with already. And as I mentioned, you can easily calculate scores for genomic regions in a very easy way using the tidyverse set of packages. All right, so let me see. I might have to stop sharing for just a second.
Stephanie, can you send us the link?
Oh, sure.
To put it in the chat.
Of the slides or of the demo?
Of the demo.
Yep, okay, I can do that.
Yeah, if you can, yeah, both please.
Okay, sure.
Yeah,
we'll do that
actually
right now.
it's from the chat. They said they want them both. Thank you.
Okay. And let me go back and now share my screen again. Okay, are you guys seeing an RStudio screen?
Yes.
Okay. Is it big? I can't tell what it looks
Yeah,
like.
yeah,
Is it readable?
it's good.
Okay,
Yeah, yeah,
perfect.
it's really readable.
All right, so I have loaded up R, and I am going to load the two packages that I've talked about, GenomicRanges and plyranges. So this is the GRanges object that I talked about before. It's the same one where you have two ranges starting from chromosome 1 with the two scores 10 and 25. And you can see what it looks like in R. Inside of this, you'll see something called IRanges. So in the second column, there's seq names, and then there's ranges, strand, gene ID, and score. Under ranges, there's an object called IRanges, and I briefly mentioned that before. So I'm going to start working with just IRanges to make this simpler for the demo. And so I'm not going to have required information about the chromosome number. I'm not going to have gene ID, strand, or score, or anything. Just to be able to show some of the functionality inside of IRanges. All right, so I'm going to create just an IRanges object. And as I mentioned, this only requires start, end, and then automatically you get the width for free. Or you can give maybe like the start and the width, and then the end would be calculated for free. And so I have three IRanges or three ranges here that start at 1, 2, and 3 position. They end at 5, 2, and 8, and then they have a given width. So some of the functions inside of plyranges, one of them is called mutate. And here we're interested in modifying or, quote, mutating a genomic interval by altering the width. And the way it works is similar to the way dplyr works with mutate. You apply the mutate function to the IRanges object, and then you need to tell it, for example, in this case, what's the width that you want to modify it. So if I apply this function to it, you see that the start position remains the same, but now I have just added 10. I've basically fixed the start position, or the term that plyranges uses is anchor. So I've anchored the start position, and then I've said just change the end to be all 10 of width. And so the widths are now all 10. This is the same as this line 21. So the default for mutate is to always anchor any changes relative to the start position. So if I run this line of code, for example, you'll see that it's the exact same as the one above. However, you can do other things. Instead of anchoring at relative to the start position, you can, for example, anchor relative to the end position. You can imagine what would happen there. So here, the end positions are the same, the 5, 2, and the 8. But now I've made the widths all 10, but I've fixed the end of the genomic ranges. So here it's easy to quickly make widths or mutate genomic ranges in a very simple way. That's what mutate means. All right, so another verb is called stretching. So I will just put this back on the screen so we can keep in mind what the IRange is. So here, now I want to stretch the IRange object. So it's not the same thing as width, or as mutate. Here, I am going to run the object and talk about it. What I do is I pick an anchor. I can pick the start, I can pick the end, or I can pick the center. And relative to wherever I pick my anchor to be, it will stretch the region in both directions or extend the region in both directions. So here, what I'm doing...
Sorry, yes. I will interrupt you. We have a question here.
Oh, yes.
Looks like start position is always less than end position, regardless of stand. Is that right?
That's a good question. I always work with it that way, but I bet you could test that out really easily. Let's see. Yeah, so start must be less than end. Yeah, good question. Thank you. Okay, so here we are going to, okay, let me clear my screen. Ooh. Here we are going to rerun the IRange object as it was before, and then stretch the width in both directions, or extend the width in both directions. So previously the widths were 5, 1, and 6, and now I have basically added an extra 10 bases to the width, but I've centered it, I've made the change relative to the center. So the center of the genomic regions are the exact same. I've just extended the regions to be of width 10. Or 10 more, if that makes sense. So that one's, I guess, a complicated one. You can also do things like shifting left and right. So this is the original IRange. If you shift left, what you're doing is you're keeping the widths the same. You are just shifting out the genomic region 10 bases in one direction, or 10 bases in the other direction, and so forth. Hopefully that makes sense. Okay. So next, let's create another genomic ranges object, and I'm going to show another verb inside of plyranges. So here I am using a different notation to create a GRanges object. This function actually comes from plyranges. So you can start with just a traditional data frame that contains the required pieces of information, such as seq names, strand, start, and width or end. And this function will automatically create for you a GRanges object. It'll convert the data frame into a GRanges object.
Stephanie, we have a question actually. Yeah. Sorry, you probably already said this, but I missed it. Are the intervals left closed and right open? Example, are they using interpose coordinate like with the bed file, or one based like with BLAST? Sorry, I meant interbase, not interpose.
I'm gonna need to, okay, do you want, maybe I can find the question. I need to reread it, sorry.
Okay. Maybe at the end we can, yeah.
Okay, that would be great. Yeah.
Yeah.
Sorry, I want to make sure I understood the question.
Yeah.
Okay, so here I've created a genomic ranges object with 7 intervals, or 7 regions now. So we're back to genomic ranges, not IRanges. But IRanges is one part of it. And then that I randomly simulated from a uniform distribution. So it means nothing. So here I am going to apply more common functions that you might recognize from dplyr, specifically group by and summarize. So here I'm interested in grouping by strand. So whether the genomic region comes from the positive strand or the negative strand of the the genome, for example, and I'm piping that into group by. And then I can pipe that into summarize and calculate the mean score for each group. So here I get an output that says for the positive strand, the mean score was 0.45, and for the negative strand, the mean score was 0.53, which is really cool. It's amazing like how hard this would be to write just like in base R sometimes. Okay, and then the last one I think I have is filter. So here I am filtering by a group now. So I'm first applying a group by function again by strand. And then for within each group, I said, please return to me the max score. So give me the largest one in that group. And these are the two genomic regions with the max score, essentially. So I hope that I have convinced you that the plyranges package is really useful if you're interested in working with genomic data to make your life easier and also to make your code more human readable. I really like it. I enjoy incorporating it into my list. This is just a taste of kind of what's in it. It has a lot of functions that you may recognize from dplyr, but then there are a lot of new ones that you don't recognize that are very specific to genomics, such as the anchoring that we talked about. You have to figure out, do I want to modify genomic ranges widths based on like the start, the end, or the middle, and so forth, things like that. And that is the end of my presentation.
Thank you very much. So let's answer some questions.
Yes. Okay, where do we go?
Yeah. So, okay. Another way to ask it is with equal to end minus start if yet the system is interbase. Thank you. So nice.
Interbase. Okay. I want to make sure I understand what you mean by interbase. Like a bed file or
one base like
a blast. Oh, you're going to make me think about this. Let's see. They start at 1, I believe. Is that what the question is?
Yeah, I think maybe 0 based or 1 based. Is it like that, or am I wrong?
I think it starts at 1 base. I think I could be wrong, and this could be embarrassing, but I'm pretty sure it's 1 base.
Okay. Okay, so we have another question. Do you normally add counts in the meta info to work with plyranges?
Great question. Right. So what I typically do is work with a different object called a summarized experiment object. And at the core, there are these two matrices that I talked about in the middle of my presentation, the counts matrix and then the metadata matrix, or the important bookkeeping matrix, where you've got the chromosomes, the start and the end positions and things like that. So what I typically do for the counts matrix is you keep it separate than this, but whenever I want to do visualizations, then I do summaries on the count matrix, and then I put them into the GenomicRanges object, and you can do cool tricks, like you can do lots of analyses similar to the way you would do like filtering and selecting and making plots, but after doing summaries, because the counts matrix, it usually has, I mean, it could have 1 sample, it could have hundreds of samples. And so what people do is they typically store them separately, but then they do summaries, they calculate summaries, and then they input them into the GenomicRanges object. And you can, for example, make that coverage plot where you can show the number of reads mapping along a particular genome and so forth.
Okay. I think I have a question.
Yes.
Waiting for more questions. How to overlap SNPs with genes? How
to overlap SNPs? So SNPs?
Yeah, SNPs.
So I don't work with SNPs very much, but I'm almost sure that like the objects that are in Bioconductor to work with SNPs on some level are genomic ranges. The difference is a SNP is of one base pair length, unless you're talking about like an indel or an insertion or a deletion, in which it might be a larger a genomic region. But you could imagine having a genomic ranges object with just the start and the end being the same position.
Mm-hmm.
And you would be able to keep track of where the SNP came from inside of the genomic ranges object. And then you can join two genomic ranges objects. So for example, you can combine genomic ranges for a SNP analysis with genomic ranges from a gene expression analysis, and you can figure out which are the genomic regions that overlap. What are the SNPs that overlap in the regions with the gene expression genomic regions? So you can do all of that very easily with genomic ranges.
Okay.
But I work less with SNPs in my own research, so I am no means an expert in that.
Okay, thank you very much.